Okay, so now we're moving into part A of the guided inquiry lab. This is the acid-base indicator part of the lab, using this bromothymol blue, some sodium hydroxide solution, and some hydrochloric acid. So hydrochloric acid, as we know, is a strong acid. Sodium hydroxide is a strong base. And this is an acid-base indicator. One of the first things that we need to do is we need to get 2 milliliters of distilled water into a test tube. So that's approximately 2 milliliters of distilled water. We'll add that into our test tube. At the very bottom you can see here. And we will add about 5 drops of the acid-base indicator. 1, 2, 3, 4, 5. We can swirl that around. It's currently a light blue color. We know that the pH of water is somewhere around 7, so that is the color that it's going when the pH is roughly in the middle. Starting with the hydrochloric acid, adding an acid will lower the pH, and will assumably change the color. After adding 5 drops of the hydrochloric acid solution, you should be able to see that the color has changed from a bluish color to a more yellowish color. We'll then be adding the strong base, the sodium hydroxide, into there to see if the color changes anymore. We'll go a drop at a time. So you can see here now that with a large concentration with an excess of the sodium hydroxide in there, you'll notice that the color is now very, very blue, almost making the neutral one pretty green, it could be said. But now that it is extremely basic, the color of this indicator has changed to blue. In part B of this lab, we actually are using this copper 2 sulfate solution right here, and some ammonium hydroxide that's super concentrated. The ammonium hydroxide makes us have to work underneath a fume hood in this part of the lab because it's super potent, and you just want it to be sent out in the fume hood. So the first thing you need to do is you need to measure 5 milliliters of that copper sulfate solution in the test tube using a graduated cylinder here. So we'll just pipette some of this into the graduated cylinder until we get to the 5 milliliter mark. Once we have the 5 milliliters, we can add it to the test tube like this. Notice that the color is blue and it's pretty clearish right now. So now we're going to be adding this ammonium hydroxide into the test tube 1 drop at a time. You'll notice here we added a few drops and something has changed. It used to be clear, and now it's a little foamy, almost. It's pretty foamy. This tells you that you are forming that copper solution, basically shifting the equilibrium to the right as you add this ammonium hydroxide, because you can make more and more of the solution, or more and more of the complex ion. Sorry. Notice that the color is getting darker and it's getting foamier and foamier. Now we're moving into part C with the cobalt chloride solution, as well as the silver nitrate. We'll be putting 2 milliliters of cobalt chloride solution into each of the 3 test tubes that you... Okay, Now we've got our 2 milliliters into each of the 3 test tubes, as you can see here. The third one is going to be our baseline test tube. And the second one will get a few drops of the silver nitrate solution. The first one will get just a few grains of the solid cobalt chloride. As you can hopefully see here, I've scooped up just a few grains of the cobalt chloride that we need. I'll be throwing these into the first test tube, like so. In the second test tube, like we said, we'll be adding a few drops of the sodium or the silver nitrate. So we have there. We'll go 1, 2, 3, 4. Now we can swirl this one around. On the side. So the first test tube against the standard test tube. There's not a whole lot of difference, although you might be able to see this one is, especially in person, this one is actually a little bit darker than the one on the right here. But then when we compare the second test tube to the third test tube, this is obviously a much more apparent difference between the pink and the blue versus 2 shades of blue. We're now moving into part D of this lab, where we'll be using sparkling water, which is carbonated with carbon dioxide. And we'll be using this CoCl green indicator, as well as a syringe to change the pressure that is experiencing, or that the carbon dioxide water basically is experiencing. So first thing we need to do is we need to get 10 milliliters, which we'll use this graduated cylinder for, into this beaker. So I'll do that real quick. So there is about 10 milliliters, and we'll add it to our beaker here of our carbonated water, sparkling water, basically. And then the next step to do is we need to add about 20 drops of this CoCl green to our solution here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 15, 16, 17, 18, 19, 20. So we've added our 20 drops of indicator. We're now going to use our syringe here that we have to get 10 milliliters of this mixture into the syringe. You just pull it up like so. So we got our 10 milliliters of the liquid. Then need to flip it over and expel the air. The way that we expel the air, we flip it over and push it back up so that all of the air bubbles get out. You want to go just until the liquid starts to drip out like so. Then we're going to take the cap once all of the air is expelled. And we're just going to cap it. Make sure that you get a firm cap on there. So here's our 10 milliliters into our thing here. We got most of the air out as we could. So you see it's a little bluish green right now. We're going to try to change the pressure that it's in so that we know that this indicator turns green, more green when it's acidic. So the way you can change the equilibrium here is by pulling on the syringe, which decreases the pressure inside of this syringe. You should notice that it turns even greener when we decrease the pressure, like so. So for part E, we'll be using this milk of magnesia, 50 milliliters of distilled water, which we have in a 50 milliliter beaker, and this universal indicator. We also need a 250 milliliter beaker like this one, which we'll be doing the things in. So first, we need 10 milliliters of the milk of magnesia, which we can get with a pipette into our graduated cylinder. So our instructions say to add the milk of magnesia into our 250 milliliter beaker. So it's 10 milliliters of milk of magnesia, as well as 50 milliliters of distilled water. You want to swirl this around to make sure that the milk of magnesia is appropriately mixed through there. Then we're going to add 5 to 10 drops of our universal indicator here. So go 1, 2, 3, 4, 5, 6, 7, 8 is right in the middle of 5 and 10. So there you go. The milk of magnesia is actually this pretty purpley color with the universal indicator added into it. That basically just indicates that the milk of magnesia is super basic. From here we'll be adding some hydrochloric acid. So obviously an acid and super acidic into the milk of magnesia to make it less basic and record the results as we go. So we'll add 1 drop at a time. Hopefully you can see if any sort of color changes begin to happen. After 1 drop, hopefully you can see it was a little bit more blue, but then it goes back to being purple. So that's basically going from like a 9.5 to a 9.0. We'll add 2 drops this time. With 2 drops, you see how it got green there? And then again, back to the purple color. We'll add 4 drops this time. 2, 3, 4. With 4 drops of hydrochloric acid in there. The color is pretty green, almost an 8.0 on the scale. But then it gets being brought back to the 9.5-ish purpley color that you see. 4, 5, 6 this time. And with 6, you saw a faint hint of a yellowish color. It's almost a 6.0, but being brought back to the purple color.